I am Georgy Kurakin. I am a clinical resident of Tver State Medical University, and I am glad to greet all the participants and organizers of this conference and present our research report titled Insights into the cyclooxygenase phylogeny. Our research project is dedicated for studying oxylipine signaling evolution by bioinformatic methods. Oxylipines are oxidized polyunsaturated fatty acid derivatives that those are abundant in different kingdoms of life and perform cell-to-cell signaling functions in almost all multicellular eukaryotes. In vertebrates, including humans, oxylipines are mediators of inflammation. In plants, they are mediators of stress, tissue injury, and pathogen invasion. In fungi, they act as quorum sensing compounds, and in brown algae and some diatom algae, they act as pheromones. Oxylipine biosynthesis in vertebrates, including humans, starts from two main enzymes, lipoxygenase and cyclooxygenase. Lipoxygenase start a pathway leading to leukotrienes and lipoxins, and cyclooxygenase starts the pathway leading to prostaglandins. This outline is conserved in plants and fungi. They also have two main enzymes of oxylipine biosynthesis. The first is lipoxygenase, and the second is a cyclooxygenase homologue, alpha-deoxygenase in plants and PSI-producing oxygenases in fungi. This similarity raises a legitimate question: what is the origin of this universality? Experimental data on lipoxygenases and cyclooxygenase-like enzymes in bacteria could help us to answer this question, but they are very scarce. Lipoxygenases are described in some cyanobacteria, myxobacteria, and opportunistic pathogens, but cyclooxygenase-like enzyme is described only in one cyanobacterium. The functions of these enzymes are almost unknown. And the aim of our research project is to infer evolution and possible functions of oxylipine biosynthesis enzymes in bacteria and in protozoa using bioinformatic methods. We started this project from publishing a paper regarding lipoxygenases in bacteria and protozoa. Here you can see the graphical outline of this work in one picture, and you can see that we revealed two associations of lipoxygenases in bacteria and in eukaryotes: association with multicellularity and association with cross-kingdom virulence. We have published these findings in our paper in Biochemistry Moscow journal. Here you can see the reference to this paper. But the next question is, what about cyclooxygenases? What is their evolution, and what are their possible functions in bacteria? Here you can see the outline of our research methods. We started from a phylogenetic analysis, then we used additional methods to confirm our evolutionary assumptions, and the last step was the statistical analysis. As I have already spoken, we encountered with low quality and low quantity of data. Cyclooxygenase-like enzyme is described only in one cyanobacterium. Here you can see the reference to this article. This enzyme functions like our lipoxygenases, generating hydroperoxide from linoleic acid. So we had very few beacons where we can use as reference points for our phylogenetic research. Moreover, it was the big danger of long branch attraction in our trees, because eukaryotic cyclooxygenase-like enzymes form relatively long branches in our trees. We used the modified Seidel-Whiting test for long branch attraction, and one tree was identified as having long branch attraction and excluded from our analysis. All other trees confirm our principal tree that includes vertebrate cyclooxygenases, fungal enzymes, plant alpha-deoxygenases, myeloperoxidase and eosinophil peroxidase-like enzymes, and cyanobacterial enzymes, including aforementioned Nostoc punctiforme enzyme that was already experimentally characterized. Here you can see that our cyclooxygenases are clustered together with some bacterial enzymes, including the cyanobacterial enzymes, and we named this cluster as our cluster. Within our cluster, the vertebrate cyclooxygenases branch is most closely related to the cyclooxygenases of ammonia-oxidizing bacteria from the order Nitrosomonadelph. The lipoxygenases are also closely related to the eukaryotic lipoxygenases, as we have revealed in our previous paper. It raises a question: could we acquire our oxylipine biosynthesis enzymes from Nitrosomonadelph? We don't know yet, but we can say that the oxylipine metabolism in Nitrosomonadelph is the most similar to human one. Operon prediction database, OperonDB, shows that in Nitrosomonadelph, lipoxygenase and cyclooxygenase-like enzyme are located on one operon. It is consistent with the fact that in humans and other vertebrates, they work together. And if vertebrates and humans had bacterial type organization of genome, lipoxygenase and cyclooxygenase were also on one operon. It was an external validation of the placement of our cyclooxygenases in our cluster together with some bacteria. The second validation comes from available PDB data for cyclooxygenase depicted in orange and alpha-deoxygenase depicted in blue. Here we can see that catalytic residues are the same, but the placement of substrate is very dissimilar. It leads to an assumption that catalytic activity of cyclooxygenases and alpha-deoxygenases have a common origin, but the ability to bind fatty acids as substrates emerged independently in these groups of enzymes. Here we try to confirm this assumption by manual alignment of cyclooxygenase 1 active site residues with the enzyme of Aspergillus fumigatus, alpha-deoxygenase, and cyclooxygenase-like enzyme of Nostoc punctiforme. Here we can see that the most similarities is observed in the case of Nostoc enzyme. The least number of similarities is observed in the case of alpha-deoxygenases, and it is confirmed by statistical analysis performed with BLAST. This alignment confirms our assumption that alpha-deoxygenases and PSI-producing oxygenases of fungi acquired their substrate binding ability independent from our cyclooxygenases and from cyclooxygenase-like enzymes in cyanobacteria. Conversely, cyclooxygenases of vertebrates and cyclooxygenase-like enzymes of cyanobacteria seem to have common origin regarding their binding sites. Here you can see residues depicted by arrows. In cyclooxygenase, these residues form a salt bridge and a hydrogen bond with the carboxylate head of the fatty acid. You can see that in cyclooxygenase-like enzyme of cyanobacteria, one of these residues is conserved, and it leads to an assumption that the common ancestor of cyanobacterial cyclooxygenase-like enzymes and our cyclooxygenases could already bind fatty acids as substrates. To investigate this, we have modeled the experimentally characterized cyanobacterial cyclooxygenase-like enzyme based on cyclooxygenase structure, and our modeling results are consistent with this assumption. They show that the substrate binding in the both enzymes is very similar. Moreover, the aforementioned tyrosine residue could really bind the carboxylate head of the substrate in both enzymes, in cyclooxygenase and in the Nostoc cyclooxygenase-like enzyme. Thank Taking together these additional data, we can now return to our principal tree and reconstruct three possible substrate binding capacity acquisition events in alpha-deoxygenases, in fungal enzymes, and in our cluster. Our cluster seems to be the only cluster that truly contains only cyclooxygenase-like oxalipine metabolizing enzymes. Therefore, it is interesting to perform a statistical analysis of this cluster and research its distribution to different taxa, like we did in the case of lipoxygenases in our previous paper. Here we can see that the leaders of cyclooxygenase-like enzyme occurrence are cyanobacteria, predominantly multicellular cyanobacteria, and multicellular actinobacteria, and it leads to an assumption that cyclooxygenase-like enzymes, like lipoxygenases, are associated with primitive multicellularity and probably cell-to-cell signaling in bacteria. This fact strongly reinforces the hypothesis of evolutionary link between oxalipines and multicellularity that we have previously advanced in our paper about lipoxygenases. We have also concluded that, in contrast to lipoxygenases that have emerged only once, peroxidase-like oxalipine biosynthetic enzymes emerged independently at least 3 times, leading to cyclooxygenases in vertebrates, alpha-deoxygenases in plants, and PSI-producing oxygenases in fungi. Our research is not published yet and not submitted to a journal, but we are planning to do it. Now, if you want to have our data or to discuss something, you can email me to the address you see at this slide. While recording this video, I hope that I will be able to answer all your questions at the questions and answer session, but if no, here you can see our contact data to ask all questions and make all requests. Thank you for your attention.